Hi everyone, we are excited to present our topic for CHM256, which is the application of chromatography in forensic science. In this presentation, we'll take you through how chromatography, something you've probably seen in labs, plays a vital role in solving crimes. Let's dig in. Before we begin, I would like to introduce our team members. First up, we have Nur Era Farina binti Muhammad Yusuf, and then we have Nur Shahira binti Yaqub, we have Najla Inara binti Muhammad Nizam, and we have Sierra Bonaventure Masabal. Here's a quick look at what we will be covering today. First, we will give a brief introduction, followed by the theory behind chromatography, then we'll go over our methodology, share an example of results, and wrap things up with a conclusion.
Let's start with the word chromatography that is derived from the Greek words chroma means color and graphene meaning to write. Originally, it referred to color writing. So what exactly is chromatography? Chromatography is a separation technique used to separate the different components of a mixture. It works based on the principle that different substances will interact differently with two phases. One is the mobile phase, which is usually a liquid or a gas that carries the mixture through the system, and the other is the stationary phase, a solid or liquid that stays fixed in place. As the mixture moves with the mobile phase through the stationary phase, the different components move at different speeds, and this allows us to separate them.
These are the types of chromatography. First, we have paper chromatography, followed by gas chromatography, liquid chromatography, high performance liquid chromatography, thin layer chromatography, column chromatography, ion exchange chromatography, size exclusion chromatography, and lastly, we have affinity chromatography. Now let's talk about the theory. How does chromatography actually works? So basically chromatography is a method used to separate mixture. Alright, so imagine you have a sample with lots of substances in it. You will let it move through two phases. First is the mobile phase, which carries the sample, and the second one is the stationary phase, which will stay put and helps to separate the part. So each substance will move at a different speed depending on its properties. And that is how we get separation. In forensic science, this is very useful because we can figure out what kind of drug someone took, we can detect poison, we can see if a signature was forged, and then we can even find what kind of fuel was used in a fire. So basically, chromatography helped us to find the invisible evidence that criminals leave behind.
So now I will explain how chromatography is used in forensic science. Step 1. Sample collection. The first step in forensic testing is to collect the evidence. This can be blood, urine, fibers, or even ink from documents. The samples must be collected carefully using clean tools and kept it in a labeled containers to avoid contamination. For step 2, sample preparation. Before we test the samples, we need to prepare them. For biological samples like blood, we may filter or mix them with a liquid. For physical evidence like ink or cloth, is dissolved in a chemical to extract the part we want to test. For step 3, the types of chromatography we use. There are different types of chromatography used in forensic depending on the kind of substance we're testing. These include gas chromatography, HPLC or high performance liquid chromatography, thin layer chromatography, and paper chromatography. For gas chromatography, GC, gas chromatography is used for substance that can become a gas, such as drugs, alcohols, and explosive things. The sample is turned into gas and passed through a column. Each substance comes out of a different time, helping us identify what is in the mixture. Second, for the high performance liquid chromatography, HPLC. HPLC is used when the sample cannot be turned into gas, like some poisons or medicine. The sample is pushed through a column filled with a liquid. Different substances travel at a different speeds, allowing us to separate and identify them. For thin layer chromatography, TLC is a simple method for fast testing. We place a small spot of the sample on a special plate and let a solvent move it upward. Different parts of the sample move at different rates and shows up as spots. And last is a paper chromatography. Paper chromatography is mostly used for testing ink or dyes. We place a spot of ink on a piece of paper and dip it into a liquid. The liquid spreads the ink and separates its colors. This helps us check if two inks are the same or different. For step 4, analysis and results. After separating the substance, we analyze them by looking at how far they move or how long they take to come out. We compare this result with non-substance to figure out what is in the sample. Machines like mass spectrometers help to confirm the findings. And step 5 is quality control. To make sure the results are correct, we test the equipment first and use blank samples to check the contamination. We also compare with standard samples to be sure the identification is accurate.
Next, I will be presenting about some fascinating analytical techniques that are critical for identifying and analyzing various substances. There will be 3 examples that will discuss the basic principles and, importantly, their diverse applications, particularly in fields like forensic science, where the precision and accuracy are paramount. So let's start with a gas chromatography, where we call it as a GC. As you can see from the diagram, GC is a powerful separation technique that works by separating compounds based on their vitality. A sample is injected, vaporized, and then carried by an inert gas. Our carrier gas through a long thin column. So, what are its practical applications? GC can use in the toxicology to detect and quantify the presence and concentration of drugs or alcohols in a body. It also plays a vital role in arson investigation that helping to identify the type of flammables liquid used in a crime scene. Second, we have a planar chromatography. Looking at the example given, you can see the distinct separation colors in their speakers. This is illustrate how this form of a liquid chromatography operates on a flat, two-dimensional surface. We put a tiny bit of the sample on the plate and then the liquid slowly moves up to the plate that carrying the different part of sample. Because some parts move faster than others, they are spread out into different spots. Where do we particularly use this? It is secrets for checking ink, like finding fake money by looking at the ink. It is also can find dyes or certain types of drug. Lastly, HPLC or high performance liquid chromatography. The diagram provides a complex system involved in these high precision techniques that use to separate, identify, and quantify components within a mixture. Unlike GC, HPLC uses a high pressure liquid mobile phase. This solvent is pumped at a high pressure through a column packed with a specific stationary phase. the interaction between the sample components and the stationary phase along with the mobile phase cause the yield weight at the different time.
In conclusion, chromatography has proven to be an indispensable analytical technique in the field of forensic science and plays a vital role in uncovering the truth. Its ability to separate, identify, and analyze the complex mixture allows forensic experts to find critical evidence in a wide range of criminal investigations. Also, chromatography ensures that decisions are made based on scientific evidence rather than speculation. As technology advances, new developments in chromatography make it even more powerful and efficient. In summary, chromatography is not just a laboratory technique, it is a powerful technique that helps in investigation and bringing justice to those who seek for it.